Okay, everybody, welcome to the 16th episode of WBESS, the Western Evolution Biology Seminar Series. Today, I have a very special guest, Roberto Vendramin. He's a postdoc at UCL and the Francis Crick Institute in London, UK. And I say that he's a very special guest because I've come across his work a few years ago when I read one of his very insightful review papers. And today he'll be talking about his work in the intersection between cancer and evolution. And I'm very excited to learn all things related to this topic. So, Roberto, thank you for being here with me today. And the floor is yours.
Thank you, Mateus, for the kind introduction and for the invitation, and thank you to everybody who's gonna watch this. Indeed, I'm Roberto, a postdoc here in London between two institutes, and I have historically a biology background, and I'm by training an RNA biology, so I'm very interested in everything RNA related, but with a really strong side passion for evolution, which is the topic of the talk today, trying to integrate a bit the geneticist point of view with the non-geneticist point of view as well in the context of cancer, which is the model I work on. And I'm starting with a simple image that you've seen quite a few times already over this series, but on your textbooks as well. So this is a bit of what probably the most famous image from Darwin's notes. And I just put it up there as a reference, but as a starting point as well. Because as I said, I have a really strong interest in evolution, but also work on cancer. And I was trying to find the similarities between evolution at the population and species level and cancer, which we'll get into in a few minutes. But before getting into that, just want to reiterate four of the most famous principles that Darwin's postulated when talking and thinking about evolution. And there's four key concepts. It's not just these ones, but I would say that these are the most important ones when thinking about natural selection and evolution, meaning overproduction, and as a result, competition between different organisms, variation, which then can also lead to speciation if big enough variation occurs over time, but also adaptation and selection, which then shape the all of the above and determine evolution in the longer scales. And now, what I've always thought is that cancer is also an evolutionary process, and it's not just me, obviously. This is the result of well, many decades, if not hundreds of years of work of different cancer biologists that started to think about cancer with an evolutionary framework because there were many different evidence. Again, we going back to the early 20th century that evolutionary forces were at play in cancer progression, in cancer initiation progression, and all the processes in between. And what actually struck me when I was thinking about the commonalities between the two is that if you look at the picture on the left, so again, the notebooks from Darwin, with the picture on the right, which is what we often see in tumors, and that was made possible with the advent of next generation sequencing, we actually see a very similar situation, meaning that from a genetic point of view, the diversity we see in tumors echoes and mimics very closely Darwin's tree of life. And again, going back to the same principles that I showed you before, we can often see them applied even in cancer, because indeed there's a constant overproduction of cancer cells, both in space but also in time. So there's a huge proliferation, which is one of the distinctive features of cancer. We have variation. We can see subclones emerging from the same founder clone, or at least from the same ancestor, meaning again, there's different cells that can be generated throughout tumor progression with either subtle or major difference between one another. We see evidence of selection. We see clones disappearing along the way. We see clones being selected. But on top of that, we also see adaptation, because not all clones have the same fitness in different environments or in different point in time, or when different types of pressures are applied. And again, evolution is blind to the future. There's no intent in evolution, but the generation of diversity will, by almost inevitably, create or lead to the generation of clones that are either better or less suited for a particular type of situation, but that may not hold for a different situation. And now, starting from kind of those principles, what I wanted to argue, and this is going back to a review that Mateus mentioned when introducing me, I wrote a few years ago, where I and my co-authors as well argued for the use of cancer as an evolutionary model, and not just to study cancer itself, which is, I think, of great interest and importance, but also to understand human evolution and evolution at large. Because one problem we have when thinking about evolution for human beings, especially, we are faced with several different problems. The first one being the timescale, which is very difficult to face, but most importantly, we don't have enough sample size, a big enough sample size to actually see evolution at play in shorter times. However, what we can do in cancer is that we can actually observe in real time evolutionary process, and others see these time and time again. We can see evidence of clonal selection, genetic drift, adaptation. We see all the different rates of evolution, all the different possible evolutionary paths that can be taken. We can also understand how mutations, so genetic mutations, but also environmental pressures and non-mutational processes can shape fitnesses of each clone or tumors at large. And in that context, we can see how the pressure of the environment, and in the case of a tumor, we're talking about low oxygen, nutrient deprivation, challenging circumstances, mechanical stresses, or other type of pressures, but also how the presence of a tough predator, meaning the immune system, can shape the evolution of tumor cells as well, because the immune system, when working, is constantly surveilling the human body, and in theory should do the same for cancer. However, cancer can find ways to escape immune surveillance, so to escape predation, and those can also inform us on how evolution can help cancer cells, but other models as well, to evade such strong selection, but also, and most importantly for the context of this talk, how therapy, so how a strong bottlenecking forces like the addition of therapy that is meant to eliminate cancer can actually influence evolution, and how cancer cells can find ways to overcome those really harsh forces. And last but not least, and I think this is what goes back to my original idea, is that by studying cancer, we can actually understand other physiological and also pathophysiological processes, because by looking at how genes can mutate to confer a determinate a certain kind of advantage, we can actually understand the function of those genes or of those cellular components. But we can also understand development and other mechanisms that are recurrently exploited by tumor cells to grow and adapt, and also understand aging a bit better due to the fast rate of proliferation in cancer cells. And all of those questions that I had were also the interest of a big consortium I was made part of when I joined in this lab in London. And that was the TracerX consortium, mainly, which has now transitioned to the next iteration, which is called TracerX-Evo. And similarly, there was another consortium of which I'm involved, which is called PEACE, and it's the endpoint of TracerX. And the idea here is to generate or to set up cancer evolutionary studies, so to include evolutionary the evolutionary background within the study of cancer formation, initiation, progression, and how cancer can lead to patients' death. And so TracerX stands for tracking cancer evolution through therapy. And we can actually look at all the different stages of tumor evolution, in this case, in lung cancer, although we've done this and we're doing this also for renal cancer, for melanoma, and for breast cancer, but the lung arm is the biggest and the most important for our study. So we start from diagnosis. We can get samples before therapy intervention, after different types of therapies are applied. We can take primary and metastatic samples, and then we can study also the patients with rapid research autopsy, so post-mortem, which is the aim of PEACE. And all of these consortia have quite big aims. There's a lot of people involved with every single different kind of expertise you can possibly imagine, but a few different primary endpoint for all of these studies and primary interest of ours. So the first one being understanding how diversity, so how heterogeneity can affect the outcome. We can have different tumors with different populations within. How does that play out when thinking about how the patients fare? And the most important question was, can we actually find rules of how these tumors evolve? And can we then use those rules, those patterns to predict how tumors maybe evolve, tumors that we haven't studied? And to do so, we have different ways of going about it. Only a few are here, but what we're trying to do is to time different events during evolution and see whether we can find recurring patterns, understand how metastasis, the process of metastatization and colonization of different organs works, again, from a clonal and subclonal level, whether we can get better at actually monitoring tumors through space and time using different techniques, so circulating tumor DNA, but also tumor cells, to understand whether we can better see these tumor cells within the body in different locations and actually figure out what's going on when we cannot actually take a biopsy, so in a metastatic context, or when the tumor mass is too small to be detected. And whether we can also find different mechanisms of evolution, of mechanisms that lead to metastatization and also to immune escape. And so, in this context, with this study, I started thinking when writing this review at the different scales of tumor evolution, because evolution is a big word and can mean many different things. And I decided to break it down in small different pieces, and I'm gonna go through each one of them over the course of the talk, starting from the most simple mechanism, so individual mutations, so small genetic alterations, and then progressively getting wider, so with bigger alterations, genetic alterations, to non-genetic mechanisms, as well to micro and macro environmental changes, and then to how this can shape how tumors can evolve. And so I'll start again going back to Darwin, because he was the first one to propose how natural selection takes small, gradual steps. And all of these small variations will eventually lead to bigger variations and potentially to speciation given enough time. It was very adamant in that evolution could only take small successive variations, so small steps forward or backwards, which is a concept that I will challenge, and it's one of the reasons why I wrote the review. But again, this is just a very famous example of how small yet important changes over time can lead to meaningful differences. In this case, just showing you different butterflies and how that can affect the color. And now, as I said, Darwin postulated that nature never makes jumps, and that even the biggest changes we see between different species or between different members of the same species are the result of a slow but continuous process, meaning that there's a gradual accumulation of mutations, which is the driver of change. And this is a bit the subject or the argument of the so-called neo-Darwinism, which takes a very Darwinian view and applied to the context of evolution. However, what I wanted to think about when writing this and thinking about how tumors evolve was how we can start by Darwinian principles, which we know are true, and then look at a bit at the broader picture. So not only how Darwinian principles can be applied to tumor evolution, but also how tumor evolution informs us on both the Darwinian principles and principles that were not postulated by Darwin, and how to integrate it all to have a bit more of a comprehensive view. Because indeed, it's been known for quite some time that whereas mutations are a very important and profound source of variation, it's not the only type of variation that can occur. And indeed, in the 40s, Goldschmidt, when thinking about evolution, so not cancer evolution, but evolution at large, postulated that there are large changes or punctuated events in evolution that are caused by macro mutations. And for example, these can be chromosomal rearrangements that can result in major leaps. So what Goldschmidt suggested was the theory of punctuated equilibrium, whereas you have long periods of stasis where there are virtually no impactful mutations happening over time, interspersed by very rare and short bursts of mutation, which led potentially to rare events resulting in profound change. And these were defined as hopeful monsters, the idea of monsters being that they are completely different from their parents, if you're thinking between about parents and their progenies. When one of these huge changes would occur, it will lead to a massive difference between parents and children, although these are rare events, hence why hopeful, because in the most cases, major changes in the genome would be not viable and would not be beneficial to the organism. But in some minor yet non-relevant cases, it will lead to generation of these monsters. And this is indeed something we can also see in tumors, and I'll get a bit more into that in a sec. But where we're looking at what we call SIN, so chromosomal instability, we can see indeed that cancer cells have different number of chromosomes. So there's aneuploidy and polyploidy. But I can also see chromosomal rearrangement. So in the bottom here, you see chromosome missing arms or having mixed pieces of different chromosomes. And again, this is the result of what we define as chromosomal instability. And indeed, macroevolution uses a different method. So this is not small mutations, small nucleotide variations, but it's a complete rearrangement of pieces or whole chromosomes to create something that has never been seen before, and changing completely the chromosomal pattern. And we and others have seen this in cancer time and time again. And here I'm just going to give a few examples of some of these mechanisms. Again, there's a lot of different labs that have done fantastic work throughout the years. I won't be able to cite them all, but this has been seen all over the place, and just focusing a bit more on what we've seen with the TracerX consortium, just because I'm a bit more familiar with it, but also because I think it gives quite a broad, it allows me to give quite broad strokes on different methods and different modes of evolution in tumors. And so indeed, we can see that somatic copy number alteration, SCNAs, happen throughout many different tumors. So virtually every tumor has at least one subclone that has somatic copy number alteration, meaning alterations in the numbers of the chromosomes in a particular cell. And these events are clearly selected for. We can see these again, across many different tumors, but across different chromosomes. So some chromosomes are more prone to these kind of events, depending on what genes are there and in the context of what cancer type you're looking at. But indeed, these mechanisms are not only shared, but quite common, indicating that indeed they are important in the context of tumor evolution. And now, these are copy number alteration in the number of particular chromosomes. However, we and others have also seen bigger, more, let's say broader events, such as whole genome doubling, which, as the name suggests, is when the whole genome of a cell is doubled, so doubling the diploidy. And we've seen tumors having, in the end, even 12 copies of their entire set of chromosomes. And again, these processes are widespread throughout different cancer types, throughout patients, and they are associated with poorer prognosis. And we think that one of the reasons why this is so important for tumor cells, especially the more malignant tumors, is that by duplicating every single chromosome, then it gives the tumors more opportunity to tinker with the genome itself. Again, it gives a bit more opportunities of messing around with the genome by creating a redundant copy that could be used to prevent lethality. Because indeed, whereas up till now I told you how the copy number can increase, what we can also see often is loss of heterozygosity, so loss of particular pieces or whole chromosomes, which is not tolerated in essential genes. However, if you're duplicating the full genome, what happens instead is that you have a backup copy of the essential gene, which allows you to then get rid of the gene that may be a tumor suppressor in the case of cancer. And again, while this is spread out throughout different tumors, all of these processes, the ones I've already talked about and the ones I will talk about in the next few slides, are not equally frequent across tumors. And that's also something very important and interesting to me as to why some tumors are more prone to certain kind of alterations of genetic and non-genetic alterations than others. We see tumors favoring whole genome doubling. We see tumors favoring SCNAs and other events as well, meaning that depending on the context, depending on the cell of origin, depending on the location of the tumor, whether it's a primary or a metastasis, a different kind of event may be selected for. Again, suggesting that it's not just a process that has to do, or that these processes are not entirely and exclusively driven by tumor cells, but also by the environment in which the tumors are. And so there's a few other events which can lead to jumps in terms of evolutions, and these are called catastrophic events because of their result. Because I showed you how you can copy or have more copies of the same chromosome or lose copies of those chromosomes, duplicating the whole chromosomal complex set. But then there's also ways of actually playing around with the content of chromosomes, which is not mutations, but actually swapping piece of the same chromosome. So we have chromotripsis, which is simply put an intrachromosomal rearrangement whereby different pieces of the same chromosomes get rearranged in a completely different pattern. And there is the breakage fusion bridge cycle, BFBs, whereby by losing a telomeric piece, the chromosome, once then replicated, can fuse with its own copy and create a massive chromosome with twice as many genes. And then, yet again, since it's missing a telomeric region, go through this cycle of breaking, fusing, and creating a bridge over and over again, creating massive genomic changes. And then there's chromoplexy, which is probably the most destructive of the three processes, whereby kind of the things that you can see in chromotripsis and in the BFP cycles can work, but across chromosomes. So there's basically complete rearrangement of chromosomes between different chromosomes. Right. Now, all of these methods, and there's all of these processes, and there's quite a lot of those that I haven't had the time to mention, fall within the umbrella term of chromosomal instability, or SIN. And these are recurrent processes we can see across different tumor types. So the question is this a feature of evolution? So are tumors bound to evolve in such a way? So do tumor cells require changes that go beyond single nucleotide changes, but actually require bigger, more steep changes in evolutionary terms? And indeed, we see that these events not only lead to poorer survival for the patients, but we see them being enriched in metastasis over primary, suggesting indeed that as tumors evolve and as they need to face harsher and harsher conditions, such as the ones that the tumor cells are subjected to when forming metastasis, these cells seem to be better fit for that purpose. And as well, when we look at whole genome doubling with this score, we can indeed see that metastasis, so the M1s here, have more genome doubling than primary tumors, suggesting indeed that these processes may be needed for tumor cells to acquire either metastatic features, so the whole set of abilities that are required for this process and to survive the harsh condition of a new environment, and colonize those environments. Now, these all of these processes go against Darwin, at least in the way that they show that evolution doesn't always need to be a slow, gradual process, but it's still evidence, it is still indicating certain different, the action of different principles postulated by Darwin, such as selection, because indeed these processes are clearly selected for, and they are inherited in a linear fashion. However, there's the extra chromosomal DNAs that actually go against another one of Darwin's principles. And I will get to that in a sec, but first, let me introduce you to ecDNA. So as the name suggests, so extra chromosomal DNA, these are circular pieces of DNA there's several ways in which they can be formed, but they're basically pieces of chromosomes that circularize and are found outside of chromosomal DNA. So they're very different from chromosomes, and this gives quite a few different advantages for cancer cells. First one being that they are very easy and quick to copy, and that they're really easily accessible. So they're easy to transcribe. And then depending on the sequence that is on the ecDNA, which could be a tumor gene, it could be an enhancer, it could be a a different part of the genome, they have different ways, but we see this over and over again for oncogenes especially. And again, we see this across different tumors. This is a rather old panel. We've seen this in many different tumors, with some particular tumors showing more ecDNA than others, like glioblastoma, for some reasons. But to go back to the first point I was making, they defy another one of Darwin's principles, which was actually Mendelian principle. So these ecDNA, since they don't have a centromere like chromosomes do, are not generally segregating like chromosomes. So they are not equally distributed through in the different daughter cells, but actually can be randomly distributed across cells, as the example shows here. Again, indicating that even in this case, different cells from the same parent cell can have a different number of these molecules, which can give different, slightly different selection advantages depending on the conditions. Again, yet another way in which tumor can evolve in a less Darwinian, less linear fashion, but a more dynamic one. And then we arrive at probably what the principle that probably defined or defies a bit more Darwin's principles, because indeed we and others, and this has been debated for a while, but now it's pretty clear that in cancer we can see evidence of Lamarckian adaptation as well, which goes in parallel to Darwinian principles, but complements these as well, showing how using both genetic and selective mechanisms as well as adaptive mechanisms can increase the fitness of an organism, or in this case, of a tumor cells. Because indeed, what we and others have seen is that tumor cells are able to differentiate and de-differentiate constantly, so pass through these two stages quite quickly and quite easily without acquiring genetic mutations in the context. So always being able to go back and forth, or have the possibility of going back and forth to one of those different cell types depending on the condition and what is required. And this has been very important and quite well studied in a particular context, so in the context of response to therapy, because whilst the idea of tumor cells being able to differentiate and de-differentiate was quite known for quite a while. If we think about the conversion that epithelial cells make when trying to extravasate and reach metastasis, the so-called epithelial to mesenchymal transition, EMT, and then the opposite one's colonization as a cord, the MET, so the mesenchymal to epithelial transition. Many researchers or many studies pointed to the importance of this transition, of this plasticity, and this ability, which seems to be intrinsic for many tumor types, of adapting to different conditions in the context of drug exposure. Because when a strong selective bottleneck is applied, and it's applied to all cells in a very rapid manner, as what happens when drugs are administered, there's usually not enough time for tumor cells to acquire mutations. So either they have clones with pre-acquired or intrinsic resistance to those drugs. Otherwise, they need to find a way to quickly adapt to what's happening, waiting or biding their time until genetic evolution kicks back in. And now we go to what in my review, in the review I wrote, I call the microenvironment, although now it's a bit more blurry whether we're thinking about micro or macro environment, because indeed I was thinking when writing this at a very small scale context of whatever is in the tumor and around the tumor, but now it's becoming a bit more clear that it's a bit more complicated than that, but I'll guide you through. Because indeed, one thing that has been observed now for some 10 years or so at least, and has been clearly been proven, is that even healthy tissue displays a huge amount of somatic mutations, even in cancer-driving genes. So we see the exact same mutations that we find in tumors that we know are oncogenes and mutations that can promote tumor growth. We see the exact same mutations in a plethora of normal cells across every single human tissue. On top of that, we see that even tumors that have clear oncogenic mutation, so when there's a genetic mutation in a clear oncogene or tumor suppressors, those mutations don't lead to the formation of the of cancers across the body in all different tissues. Because when a person has a genetic mutation, let's say in the BRCA1 or the BRCA2 gene, we know that these will predispose the person to breast and ovarian cancer, but not to other cancer types, or at least with a way lower incidence, suggesting that those tissues are particularly predisposed to those cancer mutations. We see the same for other mutations like TP53 in the Li-Fraumeni syndrome. So these patients are way higher predisposition for tumor formation in many different tissues, but not in all different tissues, and so on and so forth, suggesting that different mutations have different efficacy depending on the context where they are. Another indication is that most cancers can metastasize to specific locations, and in some cases, we know why that's the case, and it's because they can go through the bloodstream or the lymphatic vessels, or there's tropism to the bone or organs nearby. So in that case, for those cases, we know it's more physical reasons, but in other cases, there's actually no clear evidence as to why certain tumors favor some locations to metastasize, suggesting that even those locations have something particularly favorable for that particular tumor growth, or the growth of a particular tumor type. And then one thing that is pretty obvious, but it bears repeating because I don't think it's been studied enough, is that cancer incidence rises with age, but it doesn't rise linearly with the number of mutation in a person. And what is becoming more and more apparent is that cancer takes advantage also of an age soma and a weakened immune system, which is what we start seeing once people reach the, let's say, 50 years old threshold, that the soma is aging rapidly and the immune system is less and less effective, suggesting that there's different things that can shape the formation and the evolution of cancer, which are not cancer intrinsic. And indeed, as I mentioned just in the previous slide, it was a beautiful paper where they sequenced normal tissues from the esophagus of several different volunteers, healthy donors. And indeed, as you can see here, hopefully all those circles are clones or clusters of cells that harbor a particular mutation. The mutations are here, so different colors meaning different mutations, and there's different genders, different ages, and whether and people that smoked or that never smoked. And indeed, you can see that the number of mutation increases linearly with age, which is kind of the y-axis. Doesn't really change between genders, at least for the esophagus, and doesn't even change too much between smokers and no smokers. Obviously, we know that smoking increases the mutational rate, but it doesn't seem, it's not like non-smokers don't have mutations in normal tissues. They have them as well, suggesting that indeed mutations in known oncogenes, as all of these are, or tumor suppressors, are found even in normal cells. So mutations alone are not sufficient for cancer initiation, and that's great news, because otherwise, looking at these pictures, everybody should have hundreds, if not thousands, of different tumors across all organs, because this has been done in the esophagus, in the lung, in the eye, in the retina. everywhere. Now, so mutations are not enough. We need to also think about the health of the tissue, the age of the tissue, and indeed, as I was suggesting before, I was mentioning before, the risk of cancers dramatically increases once people pass the 50-year-old, let's say, threshold. And now the number of mutations, and this is not here, but you can think about the number of mutations within a person to go in a linear way across time, because mutations accumulate. Those are normal. It's due to replication of DNA in normal cells, but it doesn't match the incidence of cancer. But what actually matches it is the decrease of efficiency of the immune system and in tissue integrity, but also at the same time, the increase of inflammation and of processes that could favor cancer formation. And so this was the basis of, or these were some ideas that were circulating when I joined the lab. And about that time, a paper came out suggesting that not all carcinogens are mutagenic. Because in this, I think, landmark paper, what the authors did was to take 20 different known carcinogens and give them to mice, and then see whether the mutational signatures of these carcinogens in mice. But the results were completely unexpected, and if you ask me, quite amazing, because whilst they used 20 molecules that are known to give cancer and indeed gave cancer to those mice, only 3 out of the 20 were actually increasing the mutational load in those tumors, suggesting that 17 out of 20, although they were definitely able to promote cancer, they were not acting by increasing the mutational load in these tumors, suggesting that there's something else that doesn't have to do with mutations that is at play here. And thinking about the previous evidence of mutations being pervasive in healthy tissues as well, we thought that it's maybe a combination between mutations that are pre-existing in the tissues and some agents that may actually promote tumorigenesis. And that's indeed one of the stories, the most exciting stories out of our lab in the last few years, where we showed indeed that there are substances that are mutagens, and we know a lot of them, and we know many more with the Ames test, which doesn't always apply to humans and mice, but we know pretty much all of the mutagens. But what we don't really know is what instead can promote tumor beside increasing, without increasing the mutational burden. And what we did here was to study effects of air pollution, both in mice and in humans and in cancer cells as well, showing how pollution is not a classic initiator. It doesn't promote mutations. It doesn't promote tumor formation per se. But what it does is instead to accelerate the growth of particular cells that have pre-existing driver mutations. So it looks like it's a two-step process whereby in order to have tumor formation, you need to have mutations, but then apply selection, which in this case is the promoting agent. So we've done this with pollution, but there are several different studies showing how inflammation, how particular types of stress can be promoting tumor formation, starting from a background of a mutation, which by itself does not give enough of an advantage to dead cells in baseline condition. However, when challenged with a chemical compound, an irritant, something that is inflaming the tissue, now that mutation, or the cell with that mutation, is selected. So you start applying forces of selection to promote the growth of particular clones. And indeed, so we have a two-step model. The part on the left, it's something that we can, that has been agreed upon by every researcher working in cancer. So we know that with aging, there's mutations accumulating. We know that there are environmental mutagens, so like we have UV, smoking, and other different compounds or that can promote cancer by increasing the mutational load. We know also other behavioral patterns that may influence cancer initiation, although that matter is still a bit debated at the moment. But all of these processes can create what we refer to as dormant driver mutations. So clones or individual cells that have a mutation but are not favored under those conditions. But then when selection pressures are applied, so when promoters are introduced in the system, they will force the selection of particular clones which may be more fit for those kind of situations, thereby leading to cancer formation and kind of explaining why mutations alone are not necessary or are not sufficient to promote cancer formation. And so the situation becomes a bit more complex. And I think it's about time we have the tools to start thinking about this complexity. And again, thinking about this from with evolutionary lenses, because there's several different levels that we need to think about and whereby evolution can actually act upon, because we can think in space. So different organs, different tissues can give rise to different tumors, and those different tumors will be, or the differences within those tumors will be shaped indeed by evolutionary pressures. We can start thinking about time. So how the primary tumor grows and what forces are applied when the primary tumor is growing, and how those forces change when the tumor spreads throughout the body. But then we need to think about the microenvironment as well as the macro environment. So thinking about what is usually found in the cancer, but also cells from the immune system. We now see innervation of tumors as a common process throughout different tumor types, but then even looking throughout the space again, but in this case, spatial differences within tumors. Tumors are different, and even the same tumor is different between itself. So there's inter and intra-tumor heterogeneity. And lastly, how the whole environment can shape and promote these processes. So this brings me back to the different modes of how tumors can evolve. So there's different pressures, there are different mechanisms that can shape evolution, but how do tumors evolve? And here I showed, again in the review, I wrote 4 different types of evolution. I'll talk about 3 of them briefly, because actually linear evolution is just a particular case of branched evolution. It was the first model that was proposed, but actually is the consequence of a particular type of branched evolution, which is what you can think, what Darwin showed in his sketches, what I showed you in the initial slides. Indeed, the accumulations of mutations over time with no particular mode of accumulation. Sometimes there's more mutations, sometimes there's a few less. These represent clonal sweeps. It's kind of how we think about branched evolution. So there's not much more to add there, besides the fact that we and others see this happening across different tumor types with branches all over the place. And in one of our landmark publications, we sequenced 421 different lung tumors. And as you can kind of see here, I know this is very small, we can see evidence of branched evolution across many different tumors, but we also see in a few rare cases linear evolution, again, a subset of branched evolution, showing how this process is recurrent, especially in lung cancer, but not only. We see definitely evidence of branched evolution in cancers. However, that's not the only thing we can see, because we and others as well have showed how nature can take jumps, contrary to what Darwin was thinking. And this is what I was talking about before with those chromosomal rearrangements, whereby in this case, you have long periods of stasis and then very short bursts of mutational activity. so those punctuated events. And so these are caused by all of these different processes, plus some that we can see throughout tumors. And now another mechanism of evolution, which is a bit underappreciated, but it's definitely happening in tumors, and we see this happening all the time, is the importance of neutral evolution as well, whereby the number of alteration increases linearly across time, with no particular sign of selection for particular clones with particular mutations. And again, this has been seen in cancer over and over again, different tumor types. Here I'm not bringing our examples, although we do see it as well in TRACERx, but we see clones coexisting, no particular signs of selection. It's not necessarily always the predominant model, but it's what we observe time and time again. However, one postulation that we need to make is that neutral evolution does not exclude selection, but also vice versa. And this is very important because there's historically a bit of a friction between people favoring neutral evolution as a model, people favoring branched evolution or selection as a model. I actually think that both are happening all the time simultaneously, including punctuated evolution. And here I'm just bringing two examples totally outside the cancer field, but we can think about the elephant shark, which is, I think, the slowest evolving vertebrate. We have the coelacanth as well, kind of called the living fossils. Crocodiles are pretty much the same. And if you think about sharks especially, they've been around for a long, long while. They've survived most of the, I think, 4 of the 5 massive extinctions, and they probably are very well suited for their own environment. So once you have a static habitat and lack of predation, if you're the apex predator, and you've gotten very, very close to optimal fitness, then you don't need to evolve any further, and evolution can stay still for a while. Now, what is important to think about is that, again, evolution is blind to the future. So optimal fitness in one scenario does not necessarily mean optimal fitness in another scenario. And what we do when we sequence tumors, we look at a very short snapshot. We can sequence across times, but all of these are snapshots. And if we don't vary the conditions in between, we are always looking at the same selective pressures. However, if you change the selective pressures, if these organisms or tumor cells are presented with bottlenecking events, then they will change the rule of the game. And so that's when you will start seeing non-neutral evolutionary patterns emerge. We see this in cancer cells whereby there's neutral evolution in tumors, you apply therapy, and then evolution starts again. Because again, the pressures are changing and selection starts kicking in. But if there's very little selection, that's where neutral evolution does its best. And lastly, one thing I'd like to you to think about is how all of our predictions can stop at the most recent common ancestor. So this is again thinking about a tree and how a tree can branch, and what we can see up to the bottom of the trunk of the tree. What we don't actually see is what was there before, but was pruned because of clonal sweeps. And this is, again, going back to a very famous image, and something I like to think about, how our knowledge, or how is our knowledge influenced by a survivor bias. So we see tumors, we see the mutations, we see how they have evolved up to the MRCA. However, we don't know what was there before. We don't know what tumor cells can live without, because we actually don't see those tumors clones. So one question or two questions that I'd like you to think about is which clones were not viable and why, especially. Because that can inform us on how those evolutionary pressures shape tumor evolution and how we can exploit those to actually fight tumor formation. And indeed, these are wide implications for therapy and outcome, because the same forces that allow for the generation evolution of a malignant population can also then shape how tumors can adapt to environment and therapy. And so I will quickly go through these slides to go just a minute to how conventional therapy is performed and what are the drawbacks from an evolutionary point of view, because this is what it's normally done when treating tumors. So we start treating tumors. There's a response. We don't see tumor cells again, but then they start acquiring resistance and can relapse. And we see this all over again. We see this with different tumor types, with different treatment regimens. Unfortunately, this is the case for most therapies, especially the targeted ones. And I think the challenges are multiple, and there's multiple reasons why we fail with that approach. First, once the first line of treatment is effective and the tumor is in remission, it goes into minimal residual disease, which is difficult to detect. And it's characterized by a small population of cells that are tolerant to the first treatment and are difficult to target because they're not fast cycling, they're kind of dormant. And then when the resistance starts to emerge, and we see this again for many different kind of therapies, what we often forget, is that cancer is an evolving disease and there are virtually infinite paths towards resistance. This is the case of one particular kind of mutation of treatment with many different mutations to the same treatment in different patients or within the same patients, suggesting indeed that their options for resistance are almost limitless. And since tumors or cancer is constantly evolving, what we normally do is being reactive. So we wait for resistance to emerge and we try to deal with the emergence of resistance. However, I advocate for a different approach, a different tactic, being proactive rather than reactive, because indeed, once the resistance is fully acquired, we often see pan-treatment resistance. So different tumor types, different treatments show that resistance to one line of therapy promotes or allows for resistance to different types of therapies. Again, the bottlenecking event forces tumor cells to become resistant to many different types of therapy. And that's why I actually think that the best actionable window is within the first line of treatment and how that is provided, because treatment-naive tumors are known to respond better to treatments in general. And once you have a declining populations, there is a more stressful environment, widespread cell death, no time to acquire genetic mutations because those do take time. So possibly there, it's where we have an opportunity. And there's several people that have thought about this long and hard. One first idea is to perform adaptive therapy, which is designing an evolutionary-based therapeutic strategy to control the volume of the tumor, so the tumor volume and its growth. And indeed, that would mean giving the drug at a certain dose and certain intervals to allow the tumor to shrink, but not too much, to have still a certain number of cells responding to the treatment that keep in check the ones that may not respond to the treatment, and keeping doing or applying therapy and removing therapy depending on the tumor size. Now, unfortunately, that approach doesn't seem to be working that well as of now, and that's because our models and our techniques of measuring tumor volume and tumor growth are limited still. However, there are several studies now being undertaken which show quite some promise in this approach. Another idea is to exploit collateral sensitivity, again, using evolutionary principles to guide therapeutic decisions. And in this case, it would be to exploit the mechanisms that tumor cells use to acquire resistance and then target those. Now, as I said, there is virtually a limitless number of ways that tumor can become resistant. but in some cases, it's quite well known how resistance is acquired. It's the case for some leukemias, and this approach has been shown to work there quite nicely. Unfortunately, it's not applicable to all tumor types. And another idea is instead to convert the tumor population from what it was to begin with, using a sequence of treatments to generate a small homogeneous resistant population that is eradicated after converting it indeed into this phenotype. And again, this has been done. However, it didn't show that much promise because of the fact that it is difficult to convert the whole population into an homogeneous population of cancer cells without having persistent resistant cells there either. And then another way of going about this is to prevent adaptation. And this can be done by eradicating drug tolerant cells in a genetic way or in a non-genetic way. And indeed, in a genetic way, this has been done in several different ways and shows a lot of promise. But we and others have also done this by targeting non-mutational mechanisms, showing how preventing the ways the tumor cells or the mechanisms that tumor cells exploit to acquire or to tolerate the therapy, the stress that is generated by the therapy can actually lead to meaningful responses in preclinical models and in some cases in clinical trials. And so with that, I'd like to conclude to kind of highlight some of the concepts I've told you today. So how I think it is of the utmost importance to see cancer through evolutionary lenses. So we need to study cancer evolution both through time and space across its different stages. And in order to do that, we need to think about the regionality and the heterogeneity of a tumor. We need to be able to better monitor tumor evolution in time, and liquid biopsies may be the solution, but also to have a better idea of how metastases evolve and how tumors can reach different organs, and this can be done with rapid research autopsies. But most importantly, I think we need to expand the gene and cancer cell-centric view to better understand cancer evolution the same way as we see evolution in organisms. We don't only look at organisms, but we look at the environment. The same thing needs to be done for cancers as well. So we need to take into consideration also the tumor microenvironment, how the soma can affect, how those promoting agents that I showed you before can promote tumor formation without generating mutation. And after doing all of these, we can harness and integrate this information to design evolutionary-based or to design better evolutionary-based treatments, aiming to predict and adapt to such a dynamic system. And so this is my last line, the most important one. I'd like to thank a lot of people. Unfortunately, there's a bit too many to acknowledge, so I'll just say my two labs, Charles Wanton's lab and Ken Litchfield lab. So again, UCL and the Crick, the different consortia I'm part of, so TRACERx, TRACERx-Evo, and PEACE. Most importantly, the patients and their families and their carers, and all the agencies that found our work. And I also would like to thank all of the people whose work I've used in this presentation and didn't use in this presentation, but read and appreciated quite a lot. And you all for your attention. And if you want to get in touch, you've got a few contact details here. And thank you again.
Wow, thank you, Roberto, for sharing this wealth of knowledge. I must say that during your talk, I kept thinking that there could be a course on evolution using cancer as a model system. This was very fascinating, very insightful. And I also would like to thank you for pointing out early in your talk that, Darwinian principles are not wrong, but, we're learning more and more about different evolutionary processes. And I really liked that you point that out because you might agree with me that oftentimes, particularly, the general media will pick on alternative processes that might be, called anti-Darwinian, and then they're going to, use highlights as to trash, evolution, say evolution was wrong. And I really like that you pointed out that no, I mean, we can see in cancer different ways in which evolution develops and how evolution really happens. And I do have a few questions, but before I jump into my questions, I just want to make another point. Early in your talk, you had a quote from The Origin of Species in which Darwin says something like, nature doesn't take leaps, right? She uses small steps. And it's funny because right now I'm actually reading again on The Origin of Species. But and I went through that passage a few days ago before your talk, and I wanted to make a note. I didn't make a note on the page, but here at the very last page of actually chapter 6, when Darwin talks about the difficulties of his theory, he comes back to that idea of, which I'm going to butcher the Latin. He says, natura non facit saltum, which is that
Yeah,
idea of nature doesn't take leaps, right? And
yeah.
in fact, I think, if I'm not mistaken, Thomas Huxley, who was a strong defender of Darwin, he himself told Darwin, look, you may want to not be so adherent to that idea that evolution doesn't take leaps, but Darwin was very strong on that evolution is a slow and gradual process. But as you've shown us, there are other ways in which evolution happens, and those ways are also important. So yeah, it was very
Well,
insightful. Go ahead, sorry.
no, no, I totally agree with you. And I mean, I am a Darwinian at heart. I'm here, I'm in London. There's this huge statue of Darwin in the Natural History Museum, and I love that. And indeed, it's not like different modes of evolution make Darwin's ideas wrong, actually. Well, definitely, there may be a sentence here and there which is wrong, and it's totally fine. But what it's really nice about all of those principles is that it can still be used to integrate or to better understand the newer discoveries we're making. It's just what I wanted to stress, and I'm glad you like that, is that it's not because different modes exist that what Darwin said is wrong, and vice versa. It's just a better unified theory. But yes, please go ahead with your questions.
I agree with you. And the question that I had, and I actually was going to make another comment, but now it skipped my mind. But the question that I had is that, again, going back to the idea of using cancer as a model system to understand evolution, you've mentioned, I think, twice in which you said evolution lies to the future, right? And I understand what you mean by that, but I wanted if you can expand on that, just so whoever's watching us eventually can ponder over this.
Well, you mean to evolution is blind
Oh,
to
it's
the future?
blind. Oh, blind. Yeah, sorry. Yeah, which is also
Oh,
true. Evolution is blind to the future.
because it's there. I mean, it's something that I always find difficult, and I don't know about you when writing a paper or when thinking about, in my case, cancer, is that I want to give intention to cancer, intention to evolution, but none of those forces or none of those organisms have any kind of intention, as far as I know, at least. Uh, but indeed, it's it's very important to think how evolution is just a process and something that happens mostly because of chance. It can be guided by selective pressures and external forces, but there is no intentionality in that. So there is no anticipation of what's coming next. There's just variation, which may or may not be selected for, depending on the context. So yeah, sorry, maybe it has to do with my pronunciation, I know.
No, no, don't worry about it. No, no, but I agree with you. Yeah, that's very true, right? Stochasticity is so important in evolution. In evolution itself is a historical discipline greatly because of that. But now, one thing, and you also covered that to a good extent when you showed us the differential distribution of whole genome doubling events in different cancer types, and you alluded to that, it is puzzling to see how that distribution is different, right? Do we know whether, for instance, different cancer types they create an environment that is more prone to that doubling to happen, or actually the other way around, we are seeing a selection, right? And then all cancers are equally doubling, but then some doubling events are not actually, selected for in certain cancer types. Do we know anything of that?
So we see evidence of both things happening, and I think this has been studied over the last few years. I mean, we started seeing these with and when NGS appeared in the early, well, let's say late 2000s. It's taken a bit to be picked up. Now the costs are less prohibitive than they were, but indeed we see evidence of both things happening, and I expect to see a bit more in the upcoming years. So we see that some tumors are intrinsically or inherently less prone to acquire mutations and rearrangements. Now, the problem there is that we don't really understand why. Uh, but what we can clearly see as well is how the tumor microenvironment is actually shaping. And in that case, it's a bit easier because if you know that some tumors are caused or promoted by some agents that are clearly mutagenic, that can clearly promote rearrangements, then it's a bit easier in those contexts. But there are definitely cases where it seems to be tumor intrinsic, but it is difficult to know. Now, what you alluded to, whether we're just not seeing the doubled one because they're not viable, is an amazing question, which I expect to be true, at least in some cases, but it's the proof of the devil because you cannot actually you cannot find out how or whether that happens to an extent, at least in patients. In models, it's a bit easier, and I know of some studies that have showed some hints of that happening, but we're not there yet.
Very interesting. Well, Roberto, again, thank you for sharing this wealth of knowledge. I'm pretty sure the viewers will be delighted. Again, I really liked that idea, which is the common the central piece of your review, the idea of we can learn so much about evolution by studying cancer. And I also like that if you will, the side effect of this is that for folks who aren't familiar with evolutionary biology, it is a very good entry point because oftentimes people, again, who aren't familiar with evolution, they might think of evolution as something that only naturalists are concerned about, right? Like, and it's important just for some cryptic species in nature. But in fact, you've shown us that no, that I mean, we are just like any other species, and our cells themselves, they are undergoing evolution and causing and inflicting damage. So yeah, thank you for that again. It was very great, and I
And
look forward to.
if I may add actually to what you just said, I think some of the best papers I've read that had to or were dealing with the concept of cancer evolution and let's say response to therapy actually use concepts that were identified in bacterial studies, so in how bacteria can evolve and how they can acquire resistance to antibiotics, and apply those concepts to cancer evolution. So I not only advocate for using cancer as a model to understand evolution, but using other models to understand how cancer evolves and vice versa. It would be a bit easier if, as you said, there could be this kind of cross-pollination, people studying evolution, thinking about cancer and other diseases, other models, and vice versa, people that generally don't think about evolution, starting to get through the cancer field, which may be a bit easier, and think about those concepts and contribute to those fields as well, which is why, as I told you before, I really appreciate the series you're doing here because you're bringing around very, very different people, but all with the same central interest and looking at the same thing from multiple different sides, and it's just beautiful. It's really nice what you've been doing here.
Thank you. Thank you. I really appreciate that. Well, with that high note, I think it's a good way for us to end highlighting the whole series. Again, Roberto, thank you for your talk. And I look forward to releasing the talk in the coming days, in fact, and we'll stay in touch. Okay, I'm gonna stop
Thank
the
you so
recording
much.
now.
All
Okay.
right.